My group members and I will be presenting about the restriction enzyme. My name is Nurisma Amira binti Muhammad Noor, and I will discuss about the restriction pattern.
Hi and Assalamualaikum. My name is Shahida binti Muhammad Nawi, and I will presenting about restriction enzyme.
Hi and Assalamualaikum. My name is Nur Shafika binti Azhar, and I am going to present about Cohen and Boyer experiment and also about palindromic sequence.
Assalamualaikum everyone. My name is Nur Shazlin binti Ahmad, and I will present about why bacteria produce restriction enzyme.
Restriction enzyme were discovered and characterized in the late 1960s and early 1970s by molecular biologists Werner Abel, Hamilton O. Smith, and Daniel Nathan. Abel proposed that bacterial cells were able to protect themselves against foreign DNA through some sort of enzymatically catalyzed genetic defense mechanism. Restriction enzymes are a basic tool for biotechnology research. They are also used for DNA cloning and DNA fingerprinting. Today, scientists recognize these three categories of restriction enzyme. Type 1, which recognize specific DNA sequences but make their cut at seemingly random site that can be as far as 1,000 base pair away from the recognition site. Type 2, which recognize and cut directly within recognition site. Type 3, which recognize specific sequences but make their cut at different specific location, that is usually within 25 base pair of the recognition site. Okay, today we are going to talk about restriction enzyme. So, what is the restriction enzyme? Here we have an example that will reflect about the restriction enzyme. So, just imagine this is the bacteria and it is just floating around doing nothing, and out of somewhere here is virus, and this virus is upset today and it want to infect the bacteria. Okay, once the virus infect the bacteria, it will inject its DNA into the bacteria, and we label this with viral. Okay, so the bacteria also, being a living creature, has its own DNA. So we label this with bacteria. So basically, the virus infected the bacteria, but this bacteria will figure out some way on how to destroy this viral DNA. So there is only one way on how to destroy the viral DNA, by labeling its own DNA with these blue dots. This blue dot come from methyl group. There is an enzyme called methylase. So this methylase will methylate the bacteria DNA, and it will recognize the methylated bacteria DNA and unmethylated viral DNA. So now we have another enzyme called restriction enzyme floating around in the cytoplasm, and it will recognize the methylated DNA and unmethylated foreign DNA. So once it recognizes the unmethylated DNA, it will destroy the viral DNA.
How restriction enzymes are named? Restriction enzymes are named using the first three letters of their name from the Latin name of their source microorganism. The example that we took is Hindi tree. First letter from the genus H, which is Haemophilus. Next two letters are the first two letters of the species name in influenza. Sometimes the strain designation is include D from strain R1. If microorganism produce only one restriction enzyme and the name with Roman numeral 1.
If more than one restriction enzymes are produced, the others are numbered sequentially 2 Roman, 3 Roman, 4 Roman, and etc. Now, I will discuss about the restriction pattern. What is restriction pattern? After cutting or cleaving the DNA with a particular restriction enzyme at a particular sequence, the DNA will present in two forms or in two patterns. The pattern is called the restriction pattern. That means the restriction enzyme can cut the DNA in two ways and produce the DNA which is present in two forms or two patterns that are sticky end or blunt end. For the first pattern, which is sticky end, also called as cohesive end or staggered end, let's consider EcoR1, a common restriction enzyme used in labs. EcoR1 cuts at the following sites: 5' GAA TTC 3', 3' CTTAAG 5'. When EcoR1 recognizes and cuts this site, it always does so in every specific pattern that produces ends with single-stranded DNA overhang. If another piece of DNA has matching overhangs, which, for instance, because it has also been cut by EcoR1, the overhangs can stick together by complementary base pairing. For this reason, enzymes that leave single-stranded overhang are said to produce sticky ends. These overhangs or unpaired bases are useful during the ligation as they ensure proper joining at the fragments. For instance, a human DNA fragment can be inserted into bacterial plasmid in the right direction with the help of sticky ends. Another restriction pattern is blunt end. To explain this pattern, I take small 1 restriction enzyme as example. Blunt ends are formed by a straight cut and they do not contain any unpaired bases or overhangs at the 3' or 5' regions. Blunt-ended fragments can be joined to each other by DNA ligase. However, blunt-ended fragments are harder to ligate together, which is the ligation reaction is less efficient and more likely to fail because there are no single-stranded overhangs to hold the DNA molecules in position. Thus, blunt-ended cut require more energy than sticky-ended cut to rejoin or recombine.
What is palindromic sequence? A palindromic sequence is a nucleic acid sequence on a double-strand DNA or RNA, wherein reading 5' to 3' forward on one strand matches the sequence reading 5' to 3' on the complementary strand, which is form a double helix. Base pair of DNA are adenine, thymine, guanine, and cytosine, while for RNA do not have thymine, but RNA has uracil. Nucleotide has their own pair, which is adenine must pair with thymine for DNA, while uracil for RNA. Then guanine must be paired with cytosine. A single-stranded nucleotide sequence will be called as palindromic if it is equal to its reverse complementary. What is Cohen and Boyer experiment? They are doing copy and paste technique in 1973 until 1976. Stanley Cohen and Herbert Boyer used perfect genetic engineering to cut and paste DNA using restriction enzyme in 1973. While in 1976, they see the first expression of human gene in bacteria. In late 1960, restriction enzyme was recovered for the first time in the E. coli. The enzyme will cut at site within the foreign DNA instead of chewing from the end.
Why bacteria produce restriction enzyme? It is because bacteria, they do not have immune system as they are just simple living things. So, in order for them to survive, they need restriction enzyme to protect them against foreign DNA by cutting the DNA into pieces.
Thank you, that's all from us. We hope you are enjoying watching our video and gain some knowledge about restriction enzyme.